let's get this going. So last week we ended off here, just comparing and contrasting KCSA and KV. And then, so that hopefully we'll get your minds refreshed, we're back on the idea of transporters. And now we're going to move on to the sodium-potassium pump. So, yeah, so it's Monday. You've probably been studying, but if you haven't been, I'll just give you a few minutes to remember what the sodium-potassium pump does, and then we'll start talking about it in a couple minutes. Yeah. Yes, it is A. So, whoops. Gotta turn my pen on, I wasn't ready. Yeah, so the sodium-potassium pump's role is to reestablish ion gradients after a nerve impulse. And so, yeah, so let's talk about what it does. So, the pump moves sodium, right? How many sodium ions does it move each cycle? 3, yep, I'm seeing 3s. And does it move sodium in or out of the cell?
Sodium goes out.
Yeah, sodium goes out. And how many potassium is getting moved? 2, yep. And is potassium going in or out? Yeah, sodium and potassium is going in. So if one ion is going out and the other ion is going in, what kind of transporter is it? Uniport, symport, or antiport? Yeah, it's antiport. So with antiport, you have 2 items being moved and they're going in opposite directions, which is what we have here. For symport, you would also be moving 2 items, but they would be going in the same direction, right? And uniport would be just moving one item, one type of item. Any questions so far about how that works? All right. So, how does this transport happen? Is it random diffusion, voltage gating, ATP hydrolysis, or an ion gradient? Yeah. Yeah, exactly. So we're using ATP hydrolysis. So that makes it what kind of transport? Yeah. Yeah, primary active transport. Yeah, so the sodium-potassium pump is itself hydrolyzing ATP to accomplish this movement of ions. So that's what makes it, so it's active because we're using ATP hydrolysis, and it's primary because that pump itself is the one that's hydrolyzing the ATP to help move these ions. So, and that's different from passive mediated. How? Like, what is the biggest difference between like active transport and passive transport? Yeah, exactly. So active transport requires some kind of energy input. Passive transport is just movement of ions down their gradient. Great. And does anybody want to tell me the difference between primary and secondary types of active transport? Yeah. Yeah, exactly. So for anyone who didn't hear, so for primary active transport, it's just like we described, where that transporter itself is the one using the energy to create an ion gradient. Secondary active transport is when another transporter takes advantage of an ion gradient that's being created through another transporter using energy. So a primary active transporter might establish an ion gradient that another transporter is then using or taking advantage of for secondary active transport. Okay. So just as a review, here is that picture from the lecture of the sodium-potassium pump. So There's one really confusing or odd thing about this picture that jumped out at me when I first saw it, which is that there's actually no picture of the transporter anywhere in here, right? It's showing you all these movements, but you don't actually see the transporter. So in case anyone was confused by this, the transporter itself is embedded in the membrane. And they just, for whatever reason, I guess for clarity or not wanting to have it be too busy, they just didn't draw a picture of it. But the transporter itself is sitting within the membrane. It's a transmembrane protein. So yeah, does anybody have any questions about how this works? I can walk through it, or if someone wants to walk us through how it works, maybe that would be a good review. So, yeah. Exactly. Yeah, they're different conformations. So, yeah. So, like, let's talk about that. So, in the first step, we have sodium ion binding. The transporter is in the E1 conformation. It has ATP bound. And once ATP is bound and it's in the E1 conformation, 3 sodium ions that are inside the cell will enter, right? So we have E1 plus ATP plus 3 sodium ions. And then there is ATP hydrolysis that happens. And then we have this E1 that has that one phosphate group that is the remnant of the ATP hydrolysis, plus the 3 sodium ions. And this is what allows for a conformational change in the transporter. So it starts to open towards the outside of the cell, which is on this side, the blue colored side. And then 3 sodium ions can then exit out the other side of the cell membrane, right, on the outside of the cell. And at this point, the enzyme is the transporter is in the E2 conformation when it's open towards the outside of the cell. It still has that phosphate group attached. 2 potassium ions that are outside the cell can then enter into the transporter. And then we have phosphate hydrolysis using water. And that helps to create another conformational change from E2 back to E1. And that's what allows the 2 phosphates to then enter into the inside of the cell. And so for every one cycle, right, we're moving 3 sodium ions from the inside to the outside. We're moving 2 potassium ions from the outside to the inside. And we're hydrolyzing 1 ATP molecule. Does anybody have questions about any of that stuff? Yeah.
So this might be a double question, but it's not completely apparent to me. So okay. The KCSA, the KB, the NAB, all those channels, those are all part of the sodium potassium pump itself, right?
They are all different, yeah. Okay. So the KV and NAB work together with the sodium potassium pump to help with action potential. So we're going to go over that in the next couple slides, but KV and NAB are both going to be opening to allow sodium and potassium ions to move down their gradients. And then at the end of that, the sodium potassium pump kicks in and helps reestablish the gradients of sodium and potassium. Cool. Yeah. Do you have a question, or you're just establishing yourself? Okay. You're good. Okay. No.
So just like that word slide of like how the pump goes about.
Yeah, like sort of, so like these 2 things that we just went over, these are the sort of basic things to know. Yeah. And sort of understand that there's a conformational change that allows the transporter to be open to the inside or the outside, depending on where you are during the state of like ATP hydrolysis and everything else. But yeah, don't worry about the magnesium. Yeah. Mm-hmm. All right. Any more questions about the sodium potassium pump? Cool. All right. So now that we've talked about the sodium potassium pump, which is sort of part of this whole process, now we'll talk about the nerve impulse as a whole. And so these are also just the same slides from the lecture video, so these should look hopefully familiar to everyone. And so we have this nerve impulse involves basically like 3 important channels. So two of them are voltage gated, right? We have our KV, our potassium voltage gated channel, our NAV, our sodium voltage gated channel, and our sodium potassium pump. Each of them are going to be open or closed or inactivated at different points along the period of time for this action potential when it's happening, right? So I want everyone to take a few minutes and think about each of those channels, KV, NAV, sodium potassium pump. At which point in this diagram, when is each channel open or closed or active? And think about the movement of ions during each of those states, right, for each of those channels. So, yeah, everybody take a few minutes to think about that, and then we'll talk about it together.
I just have a question. So
Yeah.
KCSA is always open, right? And then KV is the one that's open, closed, and active.
Yes. Yeah. So, yeah, the voltage gated channels are going to be open, closed, or inactivated. The other ones are generally just open. Mm-hmm. Yeah. All right, I see some people are looking thoughtful. Some people are still writing, but maybe we should all come back together and hopefully everyone will make everything more clear. So, yeah. So I'm just gonna move over to this slide. So I just have smaller versions of the same graphs, and I put on just a little table over here, so we can talk about each of the points. I don't think it's not the table is not in your slides, but you can draw a little table if you want. So yeah, so let's start with the very beginning, right? So right here, we'll call this point A. So yeah, so let's start thinking about each of these different components. What is the membrane potential of the cell, the sodium and potassium voltage gated channels, as well as the sodium potassium pump, whether they're open or closed. And then let's think about what ions are moving and in which direction. So for point A, right at the beginning there, what is our membrane potential? That you can read from the graph, right? Is it positive or negative?
It's negative 16.
Yeah, it's negative. And so we call this polarized, right? You also can call this the resting potential. So where are our sodium and potassium ions at this point, mostly? Where is the sodium, outside or inside? Yeah, it's outside. Where is the potassium? It's inside. Excellent. So what else do we need to think about? Yeah, and let's think about our channels. So what about our sodium NAV channel? Is it open or closed or inactivated? Yeah, it's closed. Just for completeness, we'll say sodium is outside. What about our potassium voltage gated channel? Yeah, it's also closed. And our potassium is inside. What about our sodium potassium pump? Yeah. Yeah, so it's maintaining the resting potential. So yeah, so in terms of ion movement, we kind of already talked about it, right? Sodium is going outside, potassium is going in. So, all right. So let's, then things will get interesting, right? You get a nerve impulse, right, from your brain telling something to happen. And so what that results in is a little bit of a change in charge, right? So let's make our next point maybe right around here. We'll make this our point B, right when that graph is starting to go up a little bit. So what is happening here in terms of membrane potential? What's happening to the membrane? Yeah. Yeah. So it's becoming positive. And so, yeah, it's becoming more positive. So we could say it's starting to do, what's the word for that? Yeah. Depolarize, yeah. Cool. So, yeah, so that means something in terms of our voltage-gated channels, right? We're having a change in voltage in our membrane potential. So one of our channels is opening. Which channel is it that is opening? Yeah. Yeah. So the sodium channel opens. And what about our potassium channel? It still stays closed. And so, yeah, don't worry about what the sodium potassium pump is sort of doing right now in terms of member potential. This is happening really quickly, right? This has been about, a half of a millisecond or so. So what's happening in terms of ion movement? Which ions are moving and in which direction? Yeah. Which one?
Sorry, sodium's coming in.
Yeah, sodium is coming in. Great. And so when sodium is coming in, sodium is positively charged. And this is going to help make that member potential more and more positive, right? The more positive the inside is, the more positive the membrane potential is. And then eventually we reach this peak, right? Let's call this point C. So at point C, we have what is happening now with our membrane potential. Yeah. Yeah, exactly. It's most positive. It is depolarized. And so, what's happening here in terms of our voltage-gated channels? Yeah. Yeah. So the sodium channel closes, potassium channel opens. And so, so which ion is moving and where is it going? Yeah. Yeah, so potassium leaves the cell. And so when potassium is leaving the cell, potassium is also a positively charged ion, right? So if it's flowing out of the cell, what's going to happen to the membrane potential as the potassium leaves? Positive ions leaving the cell. Yeah. Yeah, it's going to become more negative, and that's exactly what we see here, right? The potential starts to go down as soon as that potassium channel opens. So, yeah, so I guess, I don't know, from this like C to D transition, or I guess we should make a point D first. So let's make a point D. And this is going to be our most negative point in terms of our membrane potential, which I just gave away, right? So our membrane potential is negative, and it's the most negative that it's going to be during this process. And so what do we call this state? Yeah. Hyperpolarization, exactly. So when we started at point A, we were polarized and negative. We went up to point C where we were the most positive and we were depolarized. And then between C and D, we became negative again. So we went from polarized down through depolarized, already way past that, to hyperpolarized. So, so what's happening during hyperpolarization? What is happening with our sodium voltage-gated channel? Yeah. It's inactivated. Yep. And what about our potassium voltage-gated channel? It's also inactivated. So now is the time when we think about our sodium-potassium pump again, right? So our voltage-gated channels are not... Whoops, did I just unplug everything? Sorry about that. All right, everything's fine. So where were we? Yeah, so the sodium potassium pump is, what is it doing right now? Yep. Yeah, exactly. So it is active. It's re-establishing the ion gradients. And membrane potential. So, not my neatest writing, but yeah, so what's happening in terms of ion movement? Which ions are moving in which direction now? Yep. Yeah. Does anybody have any questions about any of that stuff? Yeah.
I have a question about, so the way I studied it, depolarization was once you hit that, like, up to positive 40 millivolts.
Yeah.
So I was wondering why the graph shows like a span of region being like depolarized as well as like why it's specifically only up to 0.
So, yeah, that's a really good question. So yeah, it's showing depolarization as being between -60 and 0. I think, I don't know. The way I always thought about depolarization is if you're starting from polarized, which is very negative, depolarization is just the process of becoming more positive. I actually did not notice that it stopped there at 0. I would think that it would go up to positive 35 or 40, whatever that number is. Super interesting. I will look into that. Yeah. Any other questions that I may or may not know the answer to? Yeah.
So the graph at the bottom, so based off of that, NA plus, even though they both go through the cell or move like across the membrane at some point, NA plus is easier to move across, right? Because of that higher peak because the other peak is higher.
Yeah, that's an interesting question. I wouldn't say that it's necessarily easier. I would think it's just a property of the fact that they both use different transporters. So maybe the sodium voltage gated channel, like the peak is narrower, so like you have a lot of movement in a shorter amount of time comparatively. And I don't know if it could possibly, I mean, yes, like sodium and potassium are different, and sodium is smaller for one thing, and so the two channels are going to have different properties and abilities, and that's I don't know if easier is the right word for it, but like yes, it does move quicker. Yeah.
I'm confused with the y-axis show. Like, is it just
So it's showing permeability, so like the availability for sodium to be able to move across. So like it could be that there's just more voltage gated sodium channels on the membrane, and so you have just overall greater permeability because there's more of the channels. It could be because of the channel itself. I actually don't know. It could be because of the channel itself is able to move the ions across like faster. That is an interesting question that I also don't know the answer to. Yeah.
the nerve impulse causes this like reaction in the sodium and potassium ion channels, which results in an action potential that eventually like I guess like
Yeah, so it starts with like an electric impulse. So like your brain or your nerves will send electrical signals, and essentially we're showing how that electrical signal gets translated into a chemical signal through the movement of ions. Or I guess it's an electrochemical signal because ions are charged. But it's sort of that combination of electrical and chemical signaling.
and then like our action potential is the way that
Yeah, and so the action potential sort of allows that signal of charge, the electrical chemical signal, to move directionally, like down a nerve towards a target tissue, like towards a muscle, telling it to contract, or towards whatever else. Mm-hmm. Any other questions?
difference between I think I conceptually and understand but like trying to define it difference between membrane potential versus electrochemical potential.
Sure. So membrane potential is referring to the actual cell membrane. So the difference in voltage, I guess voltage is a difference, but the like the difference in charge on one side versus the other side of a cell membrane. So, and membrane potential always is from the perspective of the inside of the cell. So if membrane potential is positive, it means there's more positive charge on the inside of the membrane. Or if it's negative, there's more negative charge. For electrochemical, it's a combination. So it is partially due to charge, but it's also due to the concentration. So like this is where we're thinking more about ions, right? So like they generally like they go together, right? That's kind of how they work. Because if you had, so like let's talk about sodium ions. So you have more sodium ions outside the cell. So there's the outside of the cell will be more positive, right? And there's more sodium ions out there. And so sodium ions, both because of the difference in charge and also because of the difference in concentration of sodium itself, is going to want to go in because of the electrical and the chemical properties of that gradient. So yeah, so I'd say the difference, like one is just charge and one is both charge and concentration.
So I think why I was like specifically confused is on the slide that was defined it.
Yeah.
There was like electrical potential, chemical potential, membrane potential, and the electrochemical. So like, what's the difference of membrane versus electrical? Because in general, when we're doing this unit, we are talking about across the membrane.
Right. Yeah. So I guess like you could have an electrical potential that's across something that's not a membrane, but in the context of this class, we're only pretty much talking about membranes. But yeah, just in terms of just generally understanding an electrical potential, it doesn't have to be across a membrane, but in bodies it usually is. Yeah. Yeah. Yeah.
Speaking about different definitions, I know you explained it in the slides, but could you explain again the difference between ionophores and ion channels?
Sure. I might have to go back and review that myself, just so I don't get that wrong. Yeah, so ionophores aren't necessarily protein-based. They help to move ions, but they aren't necessarily a channel, and they aren't necessarily made of protein. Ion channels are proteins, and they go all the way through a membrane. They're like a tunnel. So like this picture of an ionophore, like it's not a channel that fully goes through a membrane. It sort of helps to carry something from one side to the other, but it's not like a channel. So like when you think of a channel, you think of just like a tunnel that passes through the membrane. Yeah. one.
Two books. So why would like the fact that the forest hydrophobic allow for rapid passage? Like I thought it would be hydrophilic.
Yeah, so it's kind of the same idea with the Multiporin. It's like that kind of like greasy slide idea where they don't want to interact with each other, and so they can sort of just like pass straight through without doing a lot of interaction with the channel itself.
So the interior is hydrophobic?
I think so. Yeah. I mean, not all of it, but a lot of it. So like the part that's very selective is not, because the water molecules interact very specifically with that part that's selective, but the rest of it is kind of just like, because it's meant for very rapid transport of water, if you want water to just kind of go straight through without interacting a lot with it, it's more hydrophobic, yeah.
And what is narrow lens?
What is what? Huh?
Like 2.8 angstroms at narrow lens.
Oh, like the most narrow point.
Oh, okay. Oh, narrow lens.
Yeah. It's all good. It's a biochem class, not an English class.
I was thinking like northwest.
Yeah, no, I could understand where you were thinking that, but sorry. Yeah, not what I meant. Just. So I guess this is maybe a good time to open it up to general exam review questions. Does anyone have questions about anything that they want to bring up in front of everyone? I also have office hours if you'd rather bring it up with me one-on-one. But if there's
Penalized for spelling.
no, if I understand what you mean, I will give you the credit.
How long's the exam?
It's like how many questions
Yeah, or like.
or how many pages?
That would be nice too.
I'll tell you each question and what it's about. I was just looking at it like yesterday. It's about, I think it's 25 or 25 pages. It's 62 questions. It's a lot of pictures. It's going to look very long, but in my opinion, it feels shorter than the first exam in that it's less writing for you. Like the first exam had several questions that a lot of people spent a lot of time writing on. This exam has less time for fewer questions where I'm asking you to write, and also like you don't have to write as many things. Like there are one or two questions where you have to write a reasonable amount, but for the most part, it's a lot more fill-in, matching, multiple choice kind of thing. Yeah, so it's longer. There's a lot of questions, but less writing. Yeah.
So a lot of diagrams I see where it seems like do we have to physically draw anything, or is it more like kind of like labeling?
There's a couple things you have to draw, not like anything major. More like drawing, curves on a graph kind of thing. No one needs artistic skills for this exam, not a requirement.
Would you recommend any specific diagrams you go over to like what we just did with the mechanism?
You do not have to draw like the action potential diagram. That's a pretty complicated diagram. No, it's a pretty simple diagram. Yeah, to understand it, I don't think it's giving away anything. There will be questions about it, but you don't have to draw it. Yeah.
How much of the NA plus glucose Zenport and the lactose permeates?
You do not need to know them.
Not at all. Or
just like a little bit. I forget what was on that slide I showed you last week. You have to know what's on there. I don't think lactate permease was on that list, was it?
I think what you were talking about, like, the list was like a little like vague and certain was just like
Okay.
understand like this versus not. So that's why we're like specific, I guess, like in this.
Yeah, for example, it's like primary versus secondary.
Yeah.
You have to know like the types of primary active transports, like the A type, P type, A type.
It wouldn't hurt to know those. Yeah, I mean, if you look at the exam review questions that are posted, that gives you a pretty good. If you can answer those I think are harder than the exam itself. So if you if you can do the exam review questions, you should be good. And that's also true in terms of content. If that content is on there, that's similar content-wise to what's in the exam. So like it's help. So like these types of transporters that are in this particular chapter, where I said like you don't have to know all of them, but like some of them is kind of vague. It's more for like understanding the concepts. Like I won't ask you specific questions about those other transporters that are not like named in that list, if that helps. Yeah. Like just understanding the basic concepts about kind of how they work. So like we were just talking about, like with Multipore, and like that's not on your list, but sort of understanding the idea of like it allows for quick movement because there's less interaction between like hydrophilic and hydrophobic things that are passing through, right? But not knowing specifically. Yeah.
How many points is this?
What's that?
How many points?
Now you're asking me hard questions. I think it's 210. Yeah. It might be like 209. I can't remember off the top of my head, but yeah, it's something around there. It's a lot of points. But they're quick points. It's like circle this. You get 2 points for circling stuff. That's good. Yeah. Yeah. Yeah.
Is the study capacity on monitor like 21?
I think so. Yeah.
Because alpha subunit has 4 domains, so it's just one alpha.
Let me go look at these notes so I don't tell you something incorrect, because where was that? Yeah. Not this. No. This. Okay.
No, like set 3 of the slides, so like more. Like it's like the right arrow.
Right.
Yeah.
This one.
No, keep going.
This one.
Keep going.
This one.
Like you have a good like 10 slides.
Okay. This is the last slide before the questions.
Oh. It's like.
I think I'm just looking at the wrong thing. I think, is it before this? No. Did I just like delete slides? Probably.
This is all like part 2. And like this is the part 3, which has like the types of primary active transport, like the glucose or the lactose permease, glucose and
Okay. Where am I?
The title of the slide is student capacity.
There we go.
This,
Oh.
Yeah.
That's it.
Yeah? Okay.
yeah.
Yeah. No? After this?
Yeah. So yeah. So
Alpha subunit. Okay. Yeah, it does say alpha subunit. So there's... I am not sure. I don't remember. Sorry. I mean, if it's not in the lecture, you don't need to know it. I can look this up and let you know. I mean, understanding like the conformational changes that you see and is more important to me than knowing like how many subunits there are. Like for the other ones, especially like where we went over like how many subunits does this one have, and especially for a lot of the porin type ones, like it's sort of more relevant. Like the fact that they're multimeric is like makes the pore, right? For this one, that's not that way. So it's there may be more than one subunit, but it's not like multiple subunits come together to form one pore, which is sort of more important in my opinion.
for PCSA,
Yeah.
like someone was asking for about the alpha form.
Yeah.
So essentially for having the hydrophobic strategy,
Yeah.
is that like because you don't want the A plus to stick to it, or you want to just have some?
Let's look at that one. So, yeah, basically, same idea. Yeah.
I have a question while we're on this topic about the difference between KCSO and KB. So KB, the big difference would be that it just has the inactivation balls, right?
Yeah, it has the whole extra piece that helps it get inactivated.
Okay.
Yeah.
But otherwise, like they're almost the same structure. They have a core.
Yeah, they're the tetramer. They have all the pieces come together to form a pore in the middle. Yeah.
And just to refresh my memory, we look at the difference between the cerebroside and the ganglioside.
Sure. Yeah, so that's from a different chapter. It's chapter 8, right? 9. Oh, right, lipids. Yeah. That is. Yeah, I actually think that the slide from the questions was a good illustration of that, if I can find where it is. Here we go. So yeah, a cerebroside has 1 sugar residue. Whoops, forgot to turn my pen on yet again. Okay, so a cerebroside, you're asking cerebroside versus ganglioside, right? Yeah, so cerebroside has just 1 sugar. Just this little green one. Gangliosides have a few sugars, oligosaccharide. That is the difference. Yeah. Yeah.
Can you go over muscle contraction?
Sure.
Like
I just had a quick question about sphingolipid.
Okay.
So is sphingolipid just a ceramide? Like I'm confused. Like how do you identify? Like what is a sphingolipid? Is it ceramide, cerebroside, ganglioside, like all of that is sphingolipid?
So yeah, I would say starting with ceramide, any of these other ones are all sphingolipids. Yeah, or I guess even a sphingosine, you could call it. No, I wouldn't. I would call this sphingosine, and I would call anything else that contains a sphingosine plus at least a fatty acid residue as a sphingolipid.
Okay, and they all would make up the
Yeah. I mean, yeah, so I know for sure in membranes you see a lot of like sphingomyelins. In terms of cerebrosides and gangliosides, I don't know if they're as common as like sphingomyelins. Yeah. Yeah, it has this. They all have this sphingosine backbone. Exactly. Yeah.
The next slide from this.
Yeah.
Would we have to do something like this on the exam?
Yes, you should be able to do this on the exam.
Okay.
Yeah.
Are we going to get like a word bank of what structural components are specific?
For some of the questions, yes, there's a word bank. For other questions, there's not a word bank.
You just have to look at them. Okay, there's my phospho.
Yeah, and like you don't have to tell me which one this is. You can just say this is a phosphate-containing head group. You don't you don't need to know the different phosphate head groups. Yeah. Oh yeah, sorry, I forgot. That is in chapter 7. Yeah.
Also, on that note.
Yeah.
No, I forgot what we did in chapter 6 and 7, so I was wondering if it's not that long ago. But do we have to know specifically like the anatomy of the myofibril? Because like Z-disc, M-disc, like all that stuff.
Not those small details. No, like more in general, like what we were talking about in terms of like the sarcomere being the functional unit. Know that like the myofibril is the muscle fiber is the cell, that kind of thing. chapter 7. Reopening. Oh, that did open. That was confusing. Okay. All right, muscle contraction. So you just want to kind of go through it, or okay, yeah. So we talked about a couple things. We talked about how it's a cycle, and so where you enter in the cycle kind of depends on the environmental state, right? So there's parts. Something important to remember is that I'm just forgetting words. It's Monday. Oh boy, the actin. Okay. Myosin heads have to bind to actin, right? And then when myosin heads move, that's they move in relative to one another. That's what causes the muscle to contract. So the important thing to consider there is that when is the myosin head able to bind to the actin, and when are those binding sites unavailable? So those binding sites are unavailable until calcium enters the cell, right? So basically, so if you're at a point when you're resting, right, and there's no calcium hanging around in the muscle cell, you are going to be basically stuck over here at this step, right, where the myosin head is no longer attached, and the next thing it would have to do is attach itself to the actin. So a helpful way to think of the cycle starting is over there, even though in the diagram it kind of looks like it starts at the top. It doesn't really start anywhere because it's a cycle, right? But when there's no calcium in the cell, it kind of pauses at that point. Once calcium enters, tropomyosin moves away. Myosin head is then able to bind weakly to actin. And the myosin also has ATP in the little binding site in the myosin head, right? So once it sticks weakly to the actin, we have release of a phosphate. And that release of phosphate helps the myosin head bind really strongly. In this, very briefly and very strongly, right, in this transient state. And then you have this power stroke where the myosin head moves, and that's going to move the myosin filament relative to the actin filament, right? Once you have that power stroke, ADP is released from that little binding site in the myosin head. Now the myosin head little nucleotide site is empty, right? And then a new ATP, because ATP is just hanging around in the cell, is going to float back into that space, and that is what is going to let the myosin head release from the actin. So now they're no longer attached to each other once that new ATP comes floating in. At that point, water helps to hydrolyze ATP, and that helps the myosin head cock, so it's ready for the power stroke again once it gets the calcium, or if there's still calcium around, we'll go through the whole cycle again. Right now it's all reset. If the calcium has left, if there's no more, it will get stuck there because the next thing it will want to do is bind to the actin. Does that help?
Oh, yeah.
Okay, cool. Yeah. Yep. Yeah. Any other questions?
I had a question from
Okay.
so, like the last question in this selected.
Let me open the sheet so I can look at what you're looking at. Sorry about the delay. I thought about trying to open everything and have it open, and then I figured my computer would probably crash if I did that, so I'm just opening them one at a time. All right, so here are the practice questions. And we're talking about the very last question. That's 22?
Yeah.
Okay.
So I understood it a certain way, but that makes sense the opposite way. Because I
Okay.
guess, can you go through the question?
Sure. So the bacterial sodium proton antiporter is a secondary active transport protein that excretes excess sodium from the cell. Is the extracellular pH higher or lower than the intracellular pH? Okay, so we have an antiporter. So let's draw maybe a little picture. Can I draw on here? No. I used to be able to draw on here. All right, let's talk through it. So, all right, we have a sodium proton antiporter, right? It's going to put sodium out. This means that if it's an antiporter, protons are coming in. If protons are coming in, what does that mean in terms of pH, right? If you have more protons, yeah, so it's going to, okay. All right, so yeah, I guess there's like more to this question. So we have to think about first sort of like what is happening in an antiporter. So when we're trying to push sodium out of the cell, right, that's going to require energy input, right? It's because normally there's already more sodium outside. So to push sodium outside, that's going to require you're pushing an ion against its gradient, right? And so the antiporter, what it's doing is it's using the movement of protons down their gradient to help drive that. So as protons are going down their gradient, that's helping to sort of provide energy to push sodium ions against their gradient. So if we know now that the protons are going down their gradient as they enter the cell, so normally there's more protons outside the cell. So if there's more protons on the outside, what does that mean in terms of the pH on the outside?
It's lower, but
yeah.
So you're talking about pH like before?
So yeah, I guess it's sort of confusing because you don't know like if it's overall gonna affect. You can think of it as just like there's such a huge excess of protons that even when this antiporter is going, it's not going to change the overall proton gradient.
Oh, so normally what is basically the gradient?
Right, and even as you allow the protons to flow down their gradient, it's not necessarily going to change it. Like there might be just so many protons outside that even when the antiporter lets some of them in as it's pushing sodium out, there's still so many more protons on the outside, and so you're not affecting necessarily the proton gradient. But that's sort of also like an assumption you have to make. Like you wouldn't necessarily know that based on this question. But yeah, I would say in general though, like these questions are harder than the exam questions. Yeah. Or you'll be given more information to make it more clear on the exam. I mean, I guess it depends on the environment, right? But I don't know. I mean, usually like the environment on the outside of a cell is, yeah, I mean, it really depends on the environment. Yeah, I don't know.
And on my note for the textbook problem section, like the first textbook problem, it just like it was asked about ethanol and whether ethanol is mediated or not mediated. How do you know by like just looking at a molecule whether it's like mediated or not mediated? Because I thought it would be mediated just because it's so pure. but it's actually not being treated?
Yeah, so it's tricky, right? So some of it is absolutely dependent on like charge or polarity. So in general, nonpolar molecules have an easier time moving through, but depending how small they are. So in ethanol's case, like it's really tiny, and so it is polar, but it's also really tiny, and so those things kind of taking both things into account, like it is able to move through. I mean, you can even think of a molecule like cholesterol that's inside the cell membranes. It does have like a hydroxyl group or two on it. So overall, it's nonpolar, but I mean, it has regions that are more polar than others. And so, since ethanol is just so tiny, it's able to go through. But yeah, I would say things to consider are size and charge, and I guess shape, but mostly size and charge. Yeah. So like something like water is also super tiny, but water is also like extremely polar. So like water is not going to want to go through. Or it does go through, but not super fast, which is why we have transporters for that. But because it's so tiny. Yeah.
Questions based off of like food protein, like anything that's COVID.
Yes. There is at least one COVID question on the exam. Mostly just the stuff that we talked about. It's not all of the stuff we talked about for COVID, but some of it is on there. Yeah. Yeah.
So related to the COVID, would there be like the spike ones with the S2 and the S1 and the elongation? Would that be on it?
It could be.
Okay.
Yeah.
I have a question about triisobutyryls.
Okay.
And I don't know if it's important to know, but like, does it matter which way we start numbering carbons?
Like on the glycerol backbone?
Yeah.
No, I don't think there's. It's symmetrical. Yeah, I don't think there's a right way or a wrong way to decide which one is 1, 2, or 3. There might be, but I don't think there is. Yeah.
Okay, even if there's like one unsaturated fatty acid and an unsaturated, even if the fatty acids are different.
So I don't think it matters as far as I'm aware. It's possible that it does, and I just don't know about it. But I mean, where it does matter is for like glycerophospholipids. But for the actual triacylglycerols, I don't think there's a right or a wrong way to number them. I'm just trying to find a picture. It's just scrolling by itself. This is great. Okay. So yeah, I mean, in this particular picture, it's showing no particular order, right? Carbon 1 on the glycerol has a mono unsaturated, carbon 2 has 2 unsaturated, polyunsaturated, carbon 3 is saturated. Like that's not in like order of saturation or anything, right? It's not an order of lengths. Maybe, yeah. Yeah, so like that's more because of the numbering. Like when you have the polar head group, the head group is on carbon 3. So that will give you I believe so, yeah. So like once you pull one off and replace it with a phosphate or a polar head group, then there's more of a directionality to it. Because, right, yeah, so carbon 3 is the one that has the phosphate containing head group, yeah.
So all the numbering really, like, I guess what would be important is like that on the third part, you would always have a phosphate.
Yeah, that is true, yep. Yeah, and just in general for numbering, you should also be able to sort of look at the fatty acid tails themselves and be able to number them and use the naming systems we talked about, like the omega system and the regular forward system and all that.
So we would be given like DECA
It'll be picture, like I'll give you like a picture, and you'll have to like look at the picture and decide things about it.
Wait, then. So like, there was like octodecanoic acid.
Okay.
I'm a bit confused because like, why would it be like why would it be octodeca to make it 18 and not a different combination of like prefixes to make that 18?
I mean, octodeca is just the prefix for 18. It's 8 plus 10.
Oh, that's just the octodeca. Is like the,
That means 18.
Okay.
Yeah. Yeah.
Would we be given just as like prefixes? Like not like the bond is on like 369, for example.
Mm-hmm.
Not that part, but like just as like prefixes because like
No, yeah, I won't ask you, like, I won't, like, give you a name of a molecule and expect you to be able to picture it in your head or draw it. It will give you pictures of molecules in general. Yeah.
So like to identify the fatty acid, we would like in terms of numbering, we would just need to know like omega, name the system, and then like numbering where the double bonds are
Yeah.
from the carboxylic acid, right?
Yeah, so, like, if you look at, like, this kind of thing, right? So you're looking at, let's uncover them, but, yeah, so just be able to tell me, you know, where the double bond is from, you know, either end of the fatty acid kind of thing.
Like should we know like the prefixes from like 14 to 18 or something like that?
Oh, no. Yeah, I won't ask you to, yeah, I'm not super interested in you knowing, like, the systematic naming. That's just sort of there in case you were interested. Yeah. Yeah. Yeah, it's more just the being able to look at the locations of double bonds, and like the numbering is more helpful because that tells you really the properties of the fatty acid. It tells you like how fluid it is, and it gives you information about, you know, relative melting point and things like that. It might be helpful to know, like, in general, in biological membranes, you have, you know, 14 to 18-ish carbons per fatty acid, but yeah, in terms of, I'm trying to think. I don't think you need to, like, if I told you stearic acid, you have to know that that's 18 carbons. I don't think I have that on there. I'm pretty sure I don't. Yeah. Do you have a question?
Yeah, all the carbons with the same amount of carbons in all the fatty acids. We do all that there's a little bit of variation in the necessity to count the amount of carbons per diagram of fatty acid.
Yeah, no, I will make it either, like, there will either be variation in length or variation in double bonds. There will not be variation in both at the same time because that's too hard to figure out just by looking at it. You'd have to actually measure it or do fancy stuff. Yeah. Any more questions? You ready? You want the exam right now? Yeah. Yeah.
Do we know the structure of fructose?
I mean, being able to recognize it and be able to identify things about it, like, is it D or L, is it alpha or beta, kind of thing, just like with any other sugar. Know that it's one of the components of sucrose, and be able to identify, like, glycosidic bonding in disaccharides, including sucrose. Any other questions? Right, yeah, know that they have different sugars added on. Like I said, you don't have to draw anything. In terms of blood types, yeah, I mean, I don't want to like give away what's on there, but yeah, no, you don't need to. Yeah, I won't really expect you to look at a lot of pictures of them. Like, we didn't really go over them in terms of like their particular structures, more in terms of just their components and like what makes you mount an immune response to them or not, that kind of thing. All right, are you all questioned out?
Any words of advice?
I guess in general, same as last time, bring a pen or pencil, bring a calculator. I guess you don't really need a calculator, but you're allowed a calculator. I don't think, you definitely don't need one, but yeah. I guess you could, I don't know. I'm trying to think if there's even a way you could try to use a calculator. There you go, you gotta do 1 + 1 + 1. Exactly, yeah. You're welcome to bring a calculator.
In terms of like studying priorities, and more studying for this exam,
Theoretically. I guess I would focus the most on the review questions that we did together in class. That will be, I think you also probably would have found that on exam 1 also. A lot of the first exam was very similar or very related to like what we mostly talked about in class. It's I mean, the stuff that's in the lectures that we didn't necessarily talk about in class is good to know, but maybe not in as much detail if you're trying to prioritize. Hopefully everyone here has started studying, but if you're not done studying. Yeah, I mean, there's lots of practice questions. I think in general the practice questions for the exam and for each chapter are more difficult than the exam itself, so if you can do those practice questions, you should be good. When I study, I like to do practice questions, so I will recommend that you do those. But you obviously don't have to. Look at the case studies if you want. A lot of them are helpful. I don't think we had any required ones. Maybe we had like 1 or 2 required ones, yeah. But look through those. Yeah. All right, well, if that is all your questions, you're free to go. I have office hours today that are, yes, there is extra credit. Yeah.